Hello, everyone, and welcome to today's webinar titled Digital Solutions to Alleviate NASH Clinical Trial Challenges. This webinar is a part of the 5th Annual Drug Discovery and Development Virtual Conference. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots. Now let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our new live chat feature during the presentation. You can find the live chat located at the right of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the bottom of your screen within the navigation bar or from the lobby. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located in the Abstract window below the Presentation window and follow the process to obtain your credits. I now present today's speakers. Paul McCracken, PhD, Vice President, Global Head of Medical Imaging, ICON, Debbie Marshall, Program Manager, ICON, and Christina Gonzalez, Project Manager, Pathology, ICON. For a complete biography on our speakers, please visit the Biography tab at the left of your screen. Paul, Debbie, and Christina, you may now begin your presentation.
Hi, thanks for joining us today. My name is Paul McCracken. I'm the Vice President and Head of Medical Imaging at ICON. I'm joined today by Tina Gonzalez and Debbie Marshall. We're presenting on the digital alternatives to alleviating NASH clinical trial challenges. Briefly, for those that aren't familiar with ICON, we're a global CRO. We provide outsourced drug and device development services, phase 1 through phase 4. We're a large organization, having acquired PRA Health Sciences last year, now some 38,000 employees. And you can see our distribution on this map here, leaders in FSP, full service phase 2-3 clinical research, early phase, late phase, real world evidence, central and specialty labs. We put together a NASH initiative within ICON, pulling together our experience and expertise. You can find more information at the link shown. Today, we're going to highlight some of our NASH expertise that we pulled together within the organization, clinical research represented by Debbie Marshall, Tina Gonzalez supporting some of the digital pathology efforts we've found, and myself for medical imaging. One of the challenges we do see within development in NASH is the probability of success. In general, for metabolic diseases, there's a very low likelihood of approval from phase 1 to marketing, some 15%. When you move to phase 2, it only improves to 25%. But specifically, since 2016, there have been 10 to 12 drug failures to make it to approval during that time, despite significant investment in that space. There are high screen failure rates, some 40 to 60%, and the FDA has asked that we target late phase disease within patient populations. So identification of those patients and showing a clinical benefit there is absolutely critical. One of the things I like to do is frame our drug development challenges within biomarkers is to look at this nice paper that was published in 2014 by AstraZeneca. They were having a relatively low success rate in their programs and tried to come up with some determinants for success within their program, and we're able to do so and boil it down to these sort of this 5R framework, right? Tissue, target, safety, patients, commercial potential. And really it describes a biomarker approach towards drug development that has proven, and they published, to improve the probability of success of their programs. And we take a similar approach to development of NASH between imaging and labs. You have coverage of a number of biomarkers that, if used appropriately, can support and improve the probability of success of drug development. Representing medical imaging, We have the leading imaging group in NASH within my department. For medical imaging at ICON, we use industry partners as well as NASH CRN radiologists for comprehensive imaging solutions that could substitute in part for invasive liver biopsies. We focus on MRI-PDFF and MRelastography. I'll describe each of those methodologies briefly today, as well as the processes around those methods that allow us to deliver the endpoints at the end of a trial. I'm sure most on the call are familiar with disease progression within NASH, where you see steatosis and fibrosis developing over time. From an imaging perspective, MRI-PDFF is able to discern fatty tissue, and that develops very early on in the disease. MRelastography, which looks at liver stiffness as a surrogate for fibrosis or eventually cirrhosis, can be developed in some of the later stages. And again, we'll describe the methodology. Very briefly, without getting into an MR physics tutorial, there are some methods developed called the Dixon methods that basically take advantage of the distinct properties between healthy tissue in the liver and fatty tissue in the liver, and by manipulating a series of pull sequences, we're able to actually get images that can show you the fat content and the water content within liver tissue, thereby being able to measure steatosis. The different vendors, GE, Siemens, and Philips, have developed sequences that do provide these maps of tissue. GE uses what's called a complex method. Philips uses what's called a magnitude method, and the Liver Lab by Siemens is a bit of a hybrid between the two. This is an example of what some of those maps look like. On the left, you can see the complex, the right magnitude, but effectively get you to the same endpoint, which is an overall distribution or map of the fat fraction derived by MRI-PDFF within the liver. Now, what's nice about these methodologies is these are longitudinal, they're non-invasive, they're not subject to the sampling bias that you see with histopathology. You're getting a 1/100,000th the volume of the liver. In fact, you're able to capture very much the entire volume of the liver and look at different regions as such. We do require that we have certain cutoffs when we enroll patients in trials. 10 is used in some trials. Sometimes it's a little bit higher, a little bit lower, depends on the sponsor and the patient population they plan to pursue. But you want to make sure you have enough of a dynamic range that when your drug does have an impact on the patient population or progression, that you're able to measure that in an accessible statistical manner. There's a lot of validation for these methodologies. These publications, in fact, are aging out. So there are a number of publications that have come subsequent to these that have, again, reinforced the validity and value of MRI-PDFF as the leading method for measuring steatosis non-invasively in the liver. Again, have been compared versus MR spectroscopy, have been compared versus histopathology, and shown to be incredibly robust and very usable in the clinical trial setting for assessment of these pathologies. This is just one example recently shown by Madrigal Pharma that has a number of assets in the NASH space, showing a dose and longitudinal dependent reduction in relative fat fraction in NASH patients as measured by MRI-PDFF. So it is being used by a number of biotech and pharma in their clinical trials. In fact, you can use imaging without biopsy up through phase 2 to assess changes in the liver and enroll patients as supported by the FDA. One of the things that we do to make sure that the assessment of MRI-PDFF of fat fraction is done appropriately and accurately is we use, with our partners, Jean Britton and her team developed at Calometrics, a phantom that can assess the scanner itself. So GE, Siemens, and Philips, you place the phantom on the left. Inside, it has a number of fat mimicking vials within it that range across the spectrum of physiologic fat fractions. We can put those in there and qualify a site to make sure they're falling within specific ranges. There's also a phantom that we use that's placed under patients in vivo in the scan that can be used to qualify and quantify that the ROIs that you're generating, that the values within the liver are in fact within an expected range. And it's an excellent QC proposition for us. In fact, this is an example that Calometrics shares But this is a case where, from the imaging perspective, the fat fraction map derived by the scanner looks okay. There's nothing, apparently problematic about it. But when you're able to assess it with the vials of fat mimicking tissue underneath, you're able to see that, in fact, there was an issue from a technology perspective on this particular patient scan, and the patient is able to be rescanned. We're able to detect this. Without the phantom in place, you would not have known this and had erroneous data potentially in your clinical trial. And just a few notes on the assessment and enrichment. So there's a few comments that we can make here and a few that have been published in this space. I won't summarize them all, but it's worth noting, certainly MRI-PDFF, it's known to be significantly more accurate than other methods for quantifying liver steatosis, and absolutely should be used in baseline and longitudinal assessments. But there are other methods that are ultrasound or transient echo based that can be used to enrich patient populations. So if you have some basic, questionnaire and then want to screen some patients with a available and less expensive tool, you might use something in that vein. And then when you start to bring patients into trials, MRI-PDFF is exquisite for that measurement. And just highlighting a few publications, and I expect this number to reduce over time, but certainly a relative reduction of 29% liver fat by MRI-PDFF has been associated and correlated well with histologic response in NASH. Non-invasive, quantitative, precise, and reproducible MRI-PDFF is emerging as a useful biomarker assessment to treat response in the setting of early phase clinical trials in NASH. Scott Reeder and team have spoken to this and published a number of times. There's also another publication, Stein et al. The meta-analysis they did it, I think it was 6 or 7 clinical trials in this space, show that, again, about a 30% relative decline in MRI-PDFF is associated with higher odds of a histologic response and NASH resolution. These support the use of MRI-PDFF in non-invasive monitoring of treatment response in early phase NASH clinical trials and provide helpful data for sample size estimation for histology-based assessments. So again, there's a wealth of information out there to support these methods in the space. Next, we have MRelastography for liver stiffness, surrogate of fibrosis. I happen to have done my training with Dick Eman at the Mayo Clinic, where my PhD was actually studying elastography, a technique I know well. And Resounded is the company that has been developed to make the technology accessible, available, reproducible. And from that, you're able to effectively put a small amplitude vibration into a patient. In this case, it's a paddle, acoustic paddle underneath the patient that then vibrates. Using certain characteristics with an MR, you're able to actually look at the phase or the waves that propagate through that tissue. That's that mental image on the bottom. And from that, you can convert those wave images to a stiffness map, and in fact, interrogate the entirety of the liver for stiffness, looking for fibrotic tissue. Again, it's important to have cutoffs in patients such that they have evaluable scans, evaluable livers, and sufficient stiffness within the liver to include them in trial and look for a dynamic change in response to therapy or progression. This map nicely illustrates, again, from, I think, Dick Eman and his team publishing, that you can see in those middle panels, and the leftmost panels, just by standard MR, you can see no difference between those livers. The middle panel, the same frequency vibration was entered into the tissue, and you can see the change in wavelength, or the difference in wavelength, across those 3 panels from top to bottom. And on the right are the elastograms, or stiffness maps, showing red being the most stiff, approximately 8 kilopascals. You can see the increasing stiffness within those levers, and that's that kind of dynamic range you're able to see. On that upper right panel, there's a correlation between MRelastography and fibrotic stage. Again, every publication that's been out there has reinforced the correlation between those methodologies with very high accuracy and reproducibility. And what's interesting to see is with the development of the 2D MRE methods and now 3D MRE methods that are becoming more popular, you're able to not only look at liver stiffness, but then some of the additional characteristics such as damping ratios are providing incremental information to that, potentially looking at the necroinflammatory, congestive, and fibrotic processes of chronic liver disease. So it's a very exciting space with significant advancements coming. And again, similar to PDFF, this is a well-validated methodology for assessment of fibrosis non-invasively, correlating to histopathology in myriad publications, and a number of publications have come subsequent to this particular reference shown below. And again, these are being used in clinical trials. The KIBA for MRE, of which I'm a participant, does have a consensus profile stating a measured change in hepatic stiffness of 19% or larger indicates a true change in stiffness has occurred with a 95% confidence. Again, Madrigal and one of the recent publications have shown using MRE, and I think this is their phase 2 trial results, a change in stiffness, and depending on how you segment the patient population to responders, highly significant changes and reductions in stiffness during the trial duration using MRelastography. So again, a superb method for non-invasively assessing changes within the liver. And I spoke to it earlier, but I just wanted to highlight with this image from the Journal of Hepatology that nicely shows that longitudinal assessment. So you're able to go back from an imaging perspective to the same region of interest over time and look for that assessment. And basically the patient acts as their own control. So you actually can do studies in a shorter duration of time with fewer patients and have, in fact, higher reproducibility or lower variability. And those two images, pairs of images in the bottom right, show the same patient at week 0 baseline and week 24, showing the relative changes in fat fraction and stiffness over time. Again, showing that really strong capability for co-localization, which is certainly important. Now, again, this reference, I'm surely has been updated, but it points out that there is a shift in the industry. That middle column speaks to phase 2 proof of concept trials that are longer than 48 weeks that have invasive liver biopsy. And you can see the numbers of those trials are small and incrementing very, very slowly. That rightmost column, in fact, shows the number of phase 2 proof of concept trials that are shorter than 28 weeks in duration that incorporate non-invasive liver measures of clinical trials. And you can see that's gone up exponentially, continues to go up exponentially. And that's an important driver to say you can make, and the regulatory agencies support this, decisions about your trials through phase 2 without doing liver biopsy. So that allows you to save biopsy for phase 3 and make sure that you've demonstrated proof of concept non-invasively in patient populations to ensure that you have chosen the right patients, that you have target engagement with your drug, chosen the right dose, and in fact see some level of efficacy with safety. So these are important non-invasive characteristics in the trend in the industry. So what can medical imaging do? Hopefully I've been able to show you a few pieces of that, but we can improve the success rates of clinical trials. You can use imaging to identify patients and certainly to stratify patients, which is absolutely important in these trials, especially in competition for patients for these trials. We've shown that there is superb correlation with histopathology, and I'll come back to that in just a moment. These can be done longitudinally for assessment of the liver. It can be done to reduce variability, and I mentioned sampling bias within the liver. These are continuous sensitive measures of change. The current scoring systems for pathology and NASH are, right, they're scoring systems. It's F1, F2, F3, F4. Whereas in fact, with imaging, you have a continuous quantitative assessment, and therefore you have an interesting approach to a statistical difference between patients that's real and meaningful. It is non-invasive. That allows you to get an early read on efficacy, potentially futility. Again, decisions are made within these trials that impact lots of patients and financially, and being able to understand how your drug's working early on is absolutely critical. Also, one of the challenges they've seen in clinical trials is the rates of disease progression varies among patients of different background and stages. And imaging can help quantify the trajectory. Again, You don't want to do a biopsy every 12 weeks or every 6 months within the same patient, but imaging you could do in theory, which allow you to look at the trajectory of different patients, responders, non-responders, etc. Coming back to histopathology, one of the things that we've been able to do, ICON is the only CRO that has imaging and labs under the same roof. And that has certainly some benefits, as those are the methodologies used for development in NASH. But what it actually allowed us to do in practice is using the PACS system for our imaging database called Mira. We were able to develop that system so that it could be used for digital histopathology. And Tina's going to speak to you next about how we've developed digital histopathology at ICON and the value therein. Thanks again for your time, and we'll answer some questions after Tina and Debbie speak.
Hi, my name is Tina Gonzalez, and I'm the ICON Central Laboratories Pathology Project Manager. As Paul has mentioned, I will be doing an overview of digital pathology. We'll be discussing the benefits of digital pathology in clinical trials, as well as the current infrastructure and workflows. There are current challenges in NASH trials, which are that biopsy samples remain to be the regulatory gold standard for efficacy assessments. However, there's significant variation in these assessments performed by local and expert hepatopathologists, which creates a need for central experts. There is also the use of consensus assessments, which require extensive time and shipping when using a glass slide approach. This leads to an environmental impact as well as a risk to slides by the transport of samples to multiple locations. Additionally, the time required to provide a result to clinicians can be large. However, there's a limited screening window. Additionally, there's an absence of currently available therapies to treat NASH and the time it takes to complete a NASH study. There are some benefits, and that would help with digital pathology. With digital pathology, there's no shipping of glass slides to expert hepatopathologists. Instead, a digital image is made available via our portal Mira. With this, shipping between facilities is not required, and shipping materials as well as courier expenses would only be needed for shipments from the site to ICON. There is also an improved turnaround time from specimen receipt to the report of results, and the average turnaround time for a multi-hepatopathologist workflow is halved. The digital images provide an option of an automated analysis via AI technologies, and there's also a reduced cost, risk of loss, and environmental impact when using digital pathology. Additionally, digital pathology can help enable creation of customizable, lean, efficient, and timely workflows, such as those listed here below. Some examples would be a single pathologist view or a multiple pathologist workflow. Additionally, there are benefits of digital pathology to our expert hepatopathologists. Our pathologists could be located anywhere in the world, but all work from the same image. The digital images would enable expert hepatopathologists to discuss examples in real time, and it increases their ability for harmonization throughout the duration of the study. Some examples of this would be facilitating regular calibration calls to optimize accuracy and avoid drift over time, and it also allows the pathologist to discuss any contentious cases. Some additional benefits of the digital tools for the pathologists are that all images are readily available, as are the list of pending cases. We can also accommodate valuable input from our key opinion leaders and implement changes in Mira. Some examples would be customizable ECRFs, customizable case notification emails, as well as the ability to view all images for a case, even if a cancellation occurs on the first set of slides. Next, we'll move on to digital pathology, infrastructure, and workflow. At ICON, we currently use a program called Mira, which is linked to the Leica AT2 as well as the AT2DX. From there, we're able to scan the whole slide images, which allows the pathologist to review them via Mira. An example of how Mira works is that the pathologist would log in, and this is how the main screen looks to a pathologist. They could then select the sponsor, protocol, queue, as well as the code, the identified code, and the corresponding barcode. If you'll note here, there is a watermark that appears when the slides are scanned on the AT2DX, and if the images are viewed on a non-medical grade monitor. From there, the combination of the AT2DX and medical grade monitor would satisfy the FDA requirements if a primary diagnosis is required. However, as we all know, primary diagnoses are not provided by a central lab, so presence of the watermark would be acceptable. Next, when logging into Mira and the case is selected, the image as well as the case report form will appear to the pathologist. At first, all of the questions would be noted in red, and when completed by the pathologist, they would move to green. Once all questions have been completed, the complete form button will then appear and allow each pathologist to sign off on their read. From there, you can see that the workflow is relatively similar to what you may expect from sample receipt, but then it changes to accommodate digital pathology. We currently can receive samples as tissue, fresh tissue, blocks, unstained slides, or even stained slides. As needed, we can stain the samples, then digitally image them. From there, the automatic images will transfer to Mira, where our laboratory team can complete an image QC and then automatically assign each case to a pathologist. Depending on how you'd like your read to be done, that we could do a primary review as read by individual pathologists. If in agreement, the case would be released to the site. However, if they're in disagreement, the case would automatically move to a consensus queue. If at consensus, the pathologist can agree, the results will be immediately reported out to the site. However, if they disagree, it would go to an adjudication queue for a read by a third pathologist. From there, the results would be finalized and reported out to sites. In summary, we've discussed the current challenges in NASH trials, such as glass slides that must be created and shipped, which risk damage to slides and impacts the environment. There's a variation in assessments performed by local and expert hepatopathologists. There's limited screening windows, but longer timelines needed to provide results, and the absence of currently available therapies and the time it takes to complete a study. There are benefits to digital pathology, such as no shipping of glass slides, which leads to reduced costs, reduced risk of loss, damage, as well as environmental impact, and it improves turnaround times from specimen receipt to reporting of results. There are additionally benefits to the pathologist when using digital pathology, and that is that the digital images allow experts to be anywhere in the world, but all work from the same image. And we can easily arrange calibration or consensus meetings to help optimize accuracy and reduce the risk of drift over time. Finally, we've discussed the digital pathology infrastructure and workflows where ICON utilizes Mira and ImageScope to collect, manage, and host digital image data, and also enable the creation of customizable lean workflows that address the needs of protocol and expert hepatopathologists. Now, if there's any questions, we can go over those at the end. And now I'll be moving on to Debbie Marshall, a program manager of clinical operations. Thank you.
Hi, my name is Debbie Marshall, like Tina mentioned. I am a program manager here at ICON in the Clin Ops area. You might wonder why I'm actually participating in this presentation since I'm not talking about digital capabilities. The point is that all these amazing technologies that Paul and Tina spoke about won't matter unless we find the right sites, as well ensure that they understand the requirements to be in a NASH study. Over the next few slides, I'll explain how we in operations support our imaging and lab partners in these complex NASH trials. So whenever I give presentations on managing NASH trials, I typically start with the challenges that they provide and how ICON can alleviate these challenges for the sites and the sponsors. Today, I'm just going to speak on this section though. Imaging and liver biopsy challenges. Requiring these procedures at this time in NASH trials will limit site and country selection. And let me caveat this by saying, if we are dealing with a site that has participated in NASH trials previously, worked with ICON imaging or labs, that's great. These challenges risks are removed. However, oftentimes, due to the competition in the NASH space, we are expanding the site identification net to bring up new sites who have NASH-NAFLD patients and are interested in getting involved. Instead of just the standard hepatology GI sites that we used to go to back in the day for NASH trials, we are now working with endocrine, family practice, internist, emergency medicine, and on and on. These are the sites that in the beginning of their NASH trial experience might not be aware of what is all required or have immediate access to like the MRI, PDFF, MRE, or biopsy. So what my team will do then is we will do a thorough feasibility at the very beginning to determine what capabilities the sites do have. Now, if they are interested in NASH and do have the patient population, but do not have the capabilities, we'll do an extra step by reaching out to the site to help them liaise with imaging or biopsy vendors. So once we have confirmed our feasibility and completed the questionnaires and all that's done, then we move forward to PSVs, right? So pre-site selection. Now, at the time of outreach for getting these PSVs scheduled, I will also attach some checklists because even though the feasibility questionnaire was filled out and all the boxes are checked, yes, 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 we have this, I just want to make sure that they truly understand what they were checking yes to. So one of these first checklists I provide is the MRI PDFF checklist. Now, you can tell by reading this that it's got technical language, so to say. So an ordinary person in the site staff would read this and not really understand it, which is the goal. We really want them to be handing this over to the imaging group to check it and make sure that they do have these requirements. So you can see here too, this one, does the scanner have the following manufacturer's liver application? For GE, it's Ideal IQ. For Philips, it's Indix and Quant. For Siemens, it's Liver Lab. If the site does answer no or an A to all of these, doesn't necessarily mean they'll be out of the study, but it means that it will require an intervention from ICON imaging to maybe help them through this process or find another sequence that works. Okay. Now, the good thing about this is we give ICON imaging a head start, right? So they can prioritize this site. The next checklist we provide is the MRE checklist. So, MRE, you either have it or you don't, right? So, because there's an application that comes from Resoundent. So this one shouldn't be that hard. However, I've had sites accidentally, check off MRE, but they really had MR enterography, not elastography. So they are two completely different things. So this is just another assurance on my side that yes, they know what we're talking about. We know we're talking about elastography, and they have that application. And finally, it's the liver biopsy checklist. So this is really to ensure that the site has thought about this requirement and have a plan in place to ensure scheduling, performing, and receiving samples will not be an issue in the trial. Okay, so now after we've completed our PSVs, or you could call them qualification visits or site selection visits or whatever you want to call them, these sites now are formally selected, right? So now they're ready to move forward with study startup, contracting, IRB submissions, and all that kind of stuff. So as soon as we have a site that is formally selected, we will send out an email stating, congratulations, you have been selected for the study. In that email, I will immediately introduce ICON imaging so they can start working on this phantom qualification, okay? Because this is an important step. You want to make sure this phantom qualification is done well in advance of SIV inactivation, because without this, you can't move forward with the study, right? So all these other tasks would be a moot point, right? So We get this going very early. If there is a problem with phantom qualification, that's going to again require ICON imaging intervention and perhaps ClinOps, because we could then go out and look for any other sites that we have local to this site that maybe can help the site participate in the study. But again, it's, we're working together to make sure this site can get activated. And then finally, after sites are up and running and participating in the study, the clinical team must continue their collaboration with imaging and labs. So you can see here, we have a standard screening window for a NASH trial. It's 8 weeks, right? Seems like a long time, but there are a lot of procedures that need to happen in here, and most often they are sequential, right? So for example, we start out with med history, con meds, vitals, EKG labs, maybe FibroScan right there in the clinic, okay? Usually takes a few days to get those labs back. So maybe depending on what they are, 3 to 5 days, I would say. After you have confirmation that these eligibility procedures have been met, then you move forward to MRI, PDFF, and MRE. Okay, but not until these are met do you want to move forward. There's no point in performing an MRE if you've got a platelet of 80, right? So then you make sure that these are performed and that they're sent in, and then ICON imaging team can analyze them and provide results. Now, I give this about 13 days. It doesn't take that long from imaging side, but the site has to perform in and send them in, right? So then after this is all done, you move forward to a liver biopsy because, oftentimes now I will see in protocols that there is a cutoff requirement in the eligibility criteria for PDFF and MRE. So let's say your PDFF is 2%, chances are they're not going to be a patient qualified to move forward to biopsy. So anyway, we go to biopsy. Biopsy does take the longest, right? Because you've got to perform it. You've got to prepare the slides or the block or the tissue. Then they've got to send it in. And then it's got to come to ICON Labs to be processed and analyzed on our end. So the point of this is there is a set length of time for all of these procedures to happen, and it takes strong teamwork between the sites and clinical and imaging and labs to make all of this work. And in summary, by us all collaborating, we get a quicker startup period. We don't exceed screening window expectations because we know where the patient is within that window, and we make sure the sequential procedures are occurring on time. And we meet the contractual timelines set forth by the contract and the client. And that's it. So now we will open up to some questions.
Thank you again, Paul, Debbie, and Christina, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until February 2024. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.